let's start here.
So today we're going to move into bacteriophages, and we're going to spend probably, my guess is 4 days on this. Okay? So that probably means it takes us through Wednesday next week. I want us to, at some point in the next week or so, discuss if we're to toss one virus out of the list that we're doing, which one do we toss? We may have to, so I just want to have that conversation. But let's start here. I'm going to turn off the lights so you can see this. But the question is, you perform a plaque assay with half a ml of filtered sewage water with E. coli O157H7 as your host. You observe the following results. First question, obviously, from this plate should be, what's the CFU per ml? Or sorry, PFU per ml. What's the concentration of bacteriophages within this sample? Okay, so take a look at it, do some counting, make sure that we're all on the same page with how to do this. There is no dilution because you took directly from half a ml of water. Thanks for asking. All right, so how do we do this calculation? What is the general format for this calculation? Perfect. So PFU over volume times dilution. Volume in this one, in this problem, half a mil, 0.5. Dilution is 1. There's no dilution to it. So it's basically just the number of PFUs divided by 0.5. How many PFUs did you count on the plate? 45? 51, 52, 50, did I hear? I think when I counted this, I counted 56, but it's a little hard on the screen, obviously, to keep track, right? The way that you would probably want to really do this in principle is take a marker and actually mark each of those plaques as you're counting to make life easier. But sometimes there are issues that we don't necessarily think about. So, for example, this one right here, how many did you count that as? I could see 3, I could see 4, I could also see 1, depending on how you define a plaque. Do the plaques have to be distinct from each other? Or do you consider the fact that it looks like, I'm going to erase that real quick. Do you count the fact that it looks like it has one right, wrong color, one right there, and then you've kind of got the outline of a second one here, outline of another one there, and an outline of another one there. These are real issues that we have when we're talking about PFUs per mil or doing these calculations. In the grand schemes, does it matter on the plate whether it's 1 versus 4? Probably not. In all honesty, the overall effect that that's going to have on your concentration that you calculate is probably rather small. But the big thing here to point out is we want these plaques to be nice and spaced out as best as possible so that we can distinguish them from each other. Would we assume from this experiment that all of those plaques are caused by the same bacteriophage?
Yes.
Okay. I don't know. You tell me. So would we assume that they're caused by the same bacteriophage?
It could be, but generally isn't every circle a different phage?
So that's true. Each plaque represents a phage that's landed down on the plate and something has happened. We'll talk about that something in a few minutes. But what else might we look for? Take a minute and just look at the plate, talk it over with somebody else, and you can argue either way, that yes, we could assume that they're all the same, or no, we could assume that they're different. Okay? So take a minute and talk it through. What might you look for?
You know it? Oh, I feel like you can't tell if these are the selective, or, you know.
So I mean, I guess infection-wise, the ones that are close together could be the same phage.
Yeah.
Top from cell to cell. The ones that are further spread out, I'd say, maybe are different.
Yeah, maybe like the different sizes too.
That makes sense.
Yeah.
Infecting more cells, the bigger the plaque.
Yeah, maybe.
Never thought of that. It's not bad.
Yeah. Micro has been a while, I feel like.
It has fun.
Yeah, I know.
All right, so what arguments could you make in favor of these all being the same phage, same exact phage infecting that lawn of bacterial cells? Can anybody make an argument for it?
I thought Grace had a decent one.
Grace, what did you say?
She was kind of thinking that like the bigger size of the plaque meant that the same phage had kind of infected that, and that's why the circle or like the circle plaque was bigger.
Okay.
So I thought that was a decent answer.
Okay. So you're focusing on size. I'm going to bet that size becomes an argument against as well, right? Why would size be an argument against?
Because different bacteriophage may have, maybe have different, I guess, what's the word, infectivity levels on the
Okay.
bacteria.
Okay. What else could we argue? Grace?
This agar isn't really like selecting for a certain phage, maybe?
That's true that it doesn't. You are selecting for phage that all have the ability to do what, though?
Lysis.
Lysis and what? Lysis of what cells?
Bacterial cells.
And more specifically, E. coli 0157H7. The PFU per mil from this sample might look completely different if we were to test against a different host strain. If we were to test against like K12 E. coli, or if we were to test against Pseudomonas fluorescens, for example. So case in point, I took sewage water a couple of weeks ago, and Amelia was trying to isolate E. coli or phage that infect E. coli 0157H7. I think, what did we find? Like 20 per mil, roughly. Mm-hmm. And for Pseudomonas, we found 100 or so phage per milliliter. So it's the number of phage that you isolate is in part dependent on what is the host strain that you're using. Just like with eukaryotic viruses, there are some viruses that can infect certain cell types and not others based on what characteristics. What defines whether a virus can infect a host cell? Receptors are a big one. And that's really a big part with viruses, is the receptors often is one of the first areas that we look at to define tropism or to define specificity of one over the other type of bacteria. Bacteriophages have what we describe as incredibly narrow host ranges. They are oftentimes, we can think of them as, do those phage infect just run-of-the-mill K12 E. coli, a general lab strain, does it infect 20 different pathogenic strains of E. coli? Can it infect Citrobacter, Shigella, ones that are all kind of related in some way to E. coli? Amelia, what did you find? Nothing. All 5 of those bacteriophage infected one strain of E. coli and no other strain that we had in our set. And that argues that tropism here is incredibly specific. And there's a lot of power to this. There's some downsides to it, but there's a lot of power to it as well when we start to think about things like phage therapy. Anybody know what that term means, phage therapy? Jonah?
It's using bacteriophages as like antibiotic, basically.
What's the downside of an antibiotic?
That resistance can occur.
Absolutely true, and resistance can occur with bacteriophages too. But if you take an oral antibiotic, that's a big issue, right? Non-specificity, which leads to oral antibiotics. If it's a broad spectrum antibiotic, you're not only killing the cells you want to, you're also destroying the rest of the microbiome, which often leads to potential secondary infections. In the hospital, we most often see things like Clostridium difficile, C. diff, right? The benefit, or at least some argue, the benefit of using bacteriophages is because they are incredibly specific to the host cell, you can come up with a targeted, quote-unquote, treatment or drug against one bacterial strain that's causing the illness and leave the rest of the microbiome uninterrupted. That's the idea. And we'll talk about this in a couple of days in one of the papers that we look at, or next week when we talk about the Pseudomonas aeruginosa and bacteriophages for it. So I just wanted to kind of put that into context to give you an idea of where this conversation, I guess, we'll focus a little bit on that in here, but that's kind of where this conversation is right now in the United States. How can we use these phages for good, okay, in a medical setting? I want to go back to the colony size real quick and just point out that one of the arguments, and this is kind of hitting on what Andrew began to say, these sizes of plaques might be dependent on things that we've already talked about. For example, burst size. How many viral particles or new bacteriophages do you get out of a single original cell that's been infected? The larger the plaque might mean larger burst size. The size of the plaque might also tell you something about how long does it take for the virus to replicate. Is it a really quick replicator that can kill a bunch of bacteria in a short period of time, or does it take 60 minutes or more for this to occur? Sometimes plaques have what's described as a bullseye pattern, where there's cells in the middle and then there's this ring outside of it of dead cells. Some have argued that is indicative that you have a temperate phage or a lysogenic phage, which is different than the lytic ones that we spend most of our time talking about. So that's how we're going to focus on all of that in the next couple of days, and that's just to kind of give you a context of what we're talking about here. Let me see if there's anything else that I wanted to do. Oh, okay. Second question down here is, how do these plaques form? So how does a plaque actually, this is a physical manifestation of a viral infection, but what leads to the formation of this plaque?
Inhibition of cell growth.
Absolutely. What else?
Lysing of the bacteriophage.
Not of the bacteriophage, but of
the
the host cell. Yes, lysis of the host cell. And by lysis, we actually mean like the explosion of that host cell, right? What else? What are the steps of an infection? So let's go through the steps of a viral infection again. What's first? Attachment.
Entry.
But do we get entry of a bacteriophage? No, it's only going to be the genomic material. So here we're going to use the term penetration instead. There's no official uncoding, since you get this physical manifestation known as a plaque, which again, as Grace was pointing out, this represents lysis. And what we ended up doing on Friday was we ended up taking one of these plaques, resuspending it in some buffered solution, and then doing serial dilutions and plating of it. And lo and behold, there are new plaques on those plates where we did serial dilutions. Okay? So this is a way to propagate or to grow those phage. Any questions on plaques? Okay. We'll come back in a minute to how these actually form, but let's shift conversation briefly for just a minute. I put up new slides for this section like a week ago, so if you've downloaded them in the past week, you're perfectly fine. But let's just start with, there are three major families of bacteriophages, known as the Myoviridae. Myo stands for, in this case, actually muscle. There are the Sipho, S-I-P-H-O, Viridae. And then there are the Podoviridae. I could write those. Can you? All right. Podo. Oops. I can't write them, apparently. Okay, so those are the three major ones. What I want you to do, I've got the pictures of these, both a cartoon image and an electron micrograph on the first couple of slides of this section. What I want you to do with a partner is take a look at those images and tell me what's unique about each of them, so how could we distinguish them from each other, and then what's common among them. And you're going to see for the Myoviridae, it's actually got structures labeled on there, so use that kind of as your guide to help you identify similarities or differences. Let's take about 5 minutes to go ahead and do this, and put together a list with your partner or partners. The Myoviridae. I don't know if that means if they're the most abundant, it's just the one we know most about. Okay. A lot of molecular studies were based on studying what's known as the T-even series of bacteriophages, and most of them are this style. Capsid shape? Yeah, and it does. Some will describe this as having both icosahedral and helical-like properties. I actually call this complex as a whole, but where the genome is primarily located is icosahedral. Most bacteriophages have what kind of genome?
Double-stranded DNA.
Yeah, most have double-stranded DNA, and we'll come back to that a little bit later. The genome size can vary, as small as 40,000 in some bacteriophage, as large as almost 200,000 in others. Okay, so these can be incredibly complex. What is the function of the tails, or these tail fibers?
For attachment.
Yeah, that's attachment. So that's what's actually binding to the host receptor. And then we've got this tube here, some sort of column in it, and then a base plate. If you were to describe the difference between the Myoviridae and then the Sipho and Podoviridae, what would you say is a major difference between them?
The length of the tail.
Yeah, that's the biggest distinguisher between them. And oftentimes what you're going to find is that on the Siphoviridae, you're not going to necessarily be able to distinguish the tail fibers as well in an electron micrograph as you might with some of the others. This tail here on the Siphoviridae is somewhat flexible. These here represent the tail fibers. The Podoviridae, on the other hand, that tube is pretty short, not very flexible. I think these are the funniest looking of all of them. All right. What are some similarities, or what are some features that they all have?
Icosahedral capsid.
Typically an icosahedral capsid. Yes, absolutely true. And that's where the genomic DNA or genomic material will be stored. What are other similarities? They've all got to have a mechanism of attachment. So they all must have some sort of tail fiber. These tail fibers are pretty weak in that they can be easily sheared just by mechanical force. Just a side note. They've all got some sort of tube or conduit to allow for the genomic material to pass from the capsid into the host cell. Okay. So all are going to have kind of a somewhat basic structure. But where they begin to differ is once you consider the penetration step. That's where there's some differences. And that's really the only difference we're going to talk about. Okay? I'm not worried about you guys focusing on the differences. I just want you to know that there are different ways in which this can happen. Okay? So appreciate the variety, I suppose, is what I'm trying to say. There we go. Oh. And there are others. These are the most abundant, but there are others as well. So there are some that would have a classical icosahedral structure, some that are helical, and then some like that infect mycoplasma. Mycoplasma have no true cell wall or peptidoglycan, so they can actually be infected by fusion events. In the same way that we can see fusion of envelope viruses in eukaryotes. And so you've got this pleomorphic or odd-shaped one over here, plasma virus, which is one that infects mycoplasma.
Are those like mycobacteria?
Mycoplasma is different than mycobacterium. They have much smaller genomes. They're actually the smallest bacteria we know of. Some of them can be pathogenic, like I think there's a Mycoplasma genitalia, which causes, I guess you could consider it a sexually transmitted infection, but there's, that's kind of the basic. So it's not Mycobacterium like tuberculosis. It's a distinct, separate bacterium. Here's the basic life cycle, lytic and lysogenic. Bacteria that perform lysogeny or a lysogenic life cycle are sometimes referred to as temperate bacteriophage. Sometimes you'll hear them referred to as lysogenic bacteriophage as well. The lytic life cycle we've already described. There are 6 steps to this. We've also talked about how this is an incredibly fast process. Note the time on here. Lytic replication at 37 degrees Celsius, for example in E. coli, might take as little as 15 minutes up to 60 minutes. This is incredibly quick. Lysogeny, on the other hand, is a rare event. Bacteria that go into lysogeny will eventually kick back into the lytic life cycle, and we'll talk about some of those decisions of how does a virus decide to go into one or the other, and what events lead into it kicking out of the lysogenic life cycle and right back into lytic life cycle. Okay, so we'll talk a little bit about those events. Ultimately, the goal for bacteriophage: get in, replicate, get out as soon as possible. So let's talk about a couple of the events here. 1, attachment. So don't even look at this picture right now, but if we're thinking about attachment of of any normal virus, what are the steps that must take place? How would we describe the attachment process?
Binding of receptors.
Okay, from what and
The
what?
binding of the tail fibers to the, I guess, cellular membrane of the bacterial cell.
Okay, and the tail fibers here are analogous to what other structure that we've talked about for just general eukaryotic viruses? Spikes perform this, or receptor binding protein. We can use that term as well. A couple of things that Amelia pointed out that I liked. You talked about a specific location. There actually can be slight differences here, whether we're talking about gram-positive or gram-negative bacteria. And I feel like what we need to do now is actually take a step back in time and review a little bit of microbiology. I know, it's so much fun. We're going to take 5 minutes to review gram-positives and gram-negative cells. Come on, guys. This is important shit. All right. I don't want to focus on gram staining or anything like that. I want to just, for a couple of moments, to focus on structure. Okay? Because, and why I want to do this is because it helps us to actually start to think about what kind of receptors would these viruses bind to. All right? That's essentially what we're going to do. So, gram-positives, you may recall, I'll start with the positives, have a really thick layer of what we call peptidoglycan. And this is also known as the cell wall. For gram-positive bacteria like Staphylococcus aureus or Streptococcus pyogenes, Bacillus megaterium, etc., this layer of cell wall can be 20 layers thick. Okay, it can be pretty dang thick. Underneath this, what we've got is a membrane. Typical phospholipid membrane. That membrane separates the outside of the cell from the inside. And so it's going to be involved in maintaining homeostasis. The other feature we may find in these gram-positive cells are molecules known as teichoic acid. And all you really need to know about them is that they're there. We use them to detect bacteria sometimes. We actually have no freaking clue what they do, but they seem to be required for bacteria to live. That's about all we know. Or I know, I suppose. So that's a gram-positive.
No cell wall.
Huh?
No cell wall.
On negatives? There is a cell wall. Well, it's thin. It's a thin cell wall, maybe 1 to 3 layers thick on our gram-negatives. What else do gram-negatives have? LPS. Where's that located, Jonah?
Kind of attached to the cell wall and then kind of sticking out.
Not from the cell wall, but they are sticking out.
From the membrane?
Outer membrane, yeah. So you've got an outer membrane. That outer membrane has on it, again, it's a phospholipid bilayer, and some of those phospholipids are modified to contain a molecule known as LPS, or lipopolysaccharide. What are we missing? Inner membrane. So gram-negative bacteria have a 2-membrane system, outer and inner, thin layer of peptidoglycan. I liken this to an Oreo. The chocolate cookie are the outer and inner membranes. The amazingly goodness of the middle is your peptidoglycan. All right. Thank you. Other things we will find, there are what's known as UMPs, or outer membrane proteins, or sometimes called porins, that are located on the outside, or located on this outer membrane. So taking a look at these two images, what then is accessible to a bacteriophage for attachment? What could it use to attach to a gram-positive cell or a gram-negative cell?
Teichoic acid for gram-positive, negative.
Okay, so teichoic acid, LPS for gram-negatives. What else?
Outer membrane protein.
UMPs or porins. On which? These are the most likely ones. Now we find on some on both of these there actually are additional ones that we haven't drawn here. Flagellin, which is a protein that makes up the flagella, could be a potential receptor. On both. We can also find proteins that make up the fimbriae as being a receptor as well, and in some rare cases even proteins that make up the pilus. But these are kind of minor ones, and they seem to be what we call reversible receptors. Ones that allow for the virus to attach to the cell, but the virus doesn't actually penetrate its DNA into the host cell when attaching to those ones. What we find primarily are the OMPs and the cell wall are the major receptors, meaning that once the virus has attached to these, it now begins a process by which it's going to penetrate and insert its DNA into the host cell. Some viruses use more than one. In fact, what we find with one of the classical T series of phage known as T4, it uses two receptors. It uses lipopolysaccharide as an initial attachment, and then it uses an outer membrane protein known as OMP-C as its secondary one, and that one leads to the irreversible binding. Oh, not yet. Okay, so here's the attachment step for a myoviridae, and one of the unique features about myoviridae is their form of penetration. Myo again means muscle. Okay, and if you think of myosin, I can't believe I am using A and P here right now, but I am. Physiology of myosin. What happens? What happens? What's that, Jonah?
It's just like movement.
Okay.
It just like bends over and pulls on.
Okay, and what would we call that? Okay, and what would we call that? Those are power strokes that lead to what?
Contraction.
Contraction. That's the key feature here. Myoviridae contract. Okay, and what you're trying to see on this, and I'll show a video of it in a minute. There's been some really exquisite studies looking at what happens when myoviridae actually attach to the host cell. So the first step is attachment of those tail fibers to the receptor. This causes the virus to basically buckle, and it kind of pushes this base plate into now the surface, or the outer membrane surface. At that point, this tail here begins to shorten in length, and as it shortens in length, it allows for this inner sheath to kind of protrude through the outer membrane. It somehow makes it through the peptidoglycan, and then it gets to the inner membrane. And it creates a conduit to allow for that genomic material to essentially diffuse out from the head. Okay. How does a virus get through how does this protein tube get through the peptidoglycan?
Lysosome.
Lysosome. Or lysozyme would be, lysosome is a eukaryotic organelle, but lysozyme is the key feature. What is lysozyme?
It's peptidoglycan hanging.
Yeah, it hydrolyzes that cell wall. This is not a slide that you have. I just found this one last night, so I'll make sure to repost these again. But viruses or bacteriophage have what we call exolysins. And this is opposed to endolysins that we'll talk about a little bit later. But broadly, what lysins are they are enzymes that break down the bonds that hold together the peptidoglycan, or the cell wall. Exolysins are ones that will be found on the actual bacteriophage viral particle and are used to help with the entry, or to help with the penetration step of the life cycle. So in this case here, what this is showing is there are 5 different major classes of exolysins. The lysozymes, which are the ones that probably, if any of them, those are the ones that we're most familiar with. Transglycosylases, glucosaminidases, and endopeptidases. The difference between these is where do they cut. And basically the key feature is they are either cutting the actual peptido, which would be these peptides, or they're cutting the glycan strands, which hold together these really long parts of the cell wall. Ultimately, though, breaks down the cell wall, allows for that protein tube to fight its way through to allow for entry of the genome into the host cell. We see this with each of these 3 viruses, that we have some sort of exolysin located on the outside there to allow it to burrow in to the cell wall and to get its DNA into the host cell. Jonah.
So would that exolysin be attached to like the base plate or
Yes.
the tube part and just like a drill bit just going?
Pretty much,
Okay.
yes. That's exactly how it works. Obviously, unlike a drill bit, you're not going to see a rotation of it, but it's basically as it's pushing down, it's chewing away or hydrolyzing those bonds to now allow it to fit through there. Exactly. Exactly how it occurs. All right, let me see if I can find this video real quick. Whoop. And we'll play this a couple of times so that you can kind of follow along and watch what's happening. But this is from a group that's actually studied this a lot. And so this is kind of a really nice rendition of the virus.
It's creepy looking.
Okay, so there's the attachment to receptors with those tail fibers. As they start to contract, look at these, like this is the thing of nightmares, right? So you get this secondary attachment,
Oh my God.
lysozyme, and notice what's happening up here. This whole thing is kind of like corkscrewing, and as it's corkscrewing, I don't know what that part represents, sorry. But
Maybe
there's that
that
dissolving the peptidoglycan and allowing for that DNA now to get into the cell. Shall we watch again?
Yes.
Follow through again? Actually, it'd be nice to play this at half speed, but I don't know how to do that.
It's okay.
The mothership is
Are those things that extend out the base plate?
Yeah. And notice how that top is
So that's
turning
all
now,
it's
and as it's doing, it's contracting. And now you've got this central sheath that's moving down.
And then what are those green? The
I think that's supposed to be the lysozyme, but I don't know why they show it as detaching. I can't totally follow the last part of this video. And I read their like little excerpt with it, and it doesn't make sense with how I understand this process to occur.
Okay.
Because that white piece was supposed to be the DNA, and that makes no sense either. Jonah.
Does the base plate need to attach to specific receptors?
You know, that's a good question. I don't know
Looks like it will.
Yeah, it does look like that, doesn't it? And I don't know the exact answer to that. It may very well have to do so, and I think that leads to what we call the irreversible binding step, in which now the virus has committed to infecting that cell. But I don't know for certain. Go ahead.
So if there were like more than one receptor that it needed to attach to, would be like that?
That would be my guess, potentially. Sabrina?
Do each of the tail fibers have to attach to a receptor?
I don't believe so. I don't know if each of them has to or not, but one of the things that I read is you need to have at least attachment with 3 of them for this commitment to take place. So I'm guessing it's kind of one of those things that the more attachment you have, the better. There's probably a critical threshold that's required. Go ahead, Jonah.
Does attachment cause like some sort of conformational change of the spikes to?
It does.
In order for it to.
Yeah, and you have to remember that this happens, all of this is happening without ATP. the viral particle itself has no energy whatsoever. So yeah, all of these events, the binding triggers some sort of conformational change as it shows in this video for that base plate to release. And once that binds, now you start to get the contraction of that tail tube as well. So you're absolutely right that all of these are basically one event is somehow leading to a conformational change of proteins in the next event and so on and so forth. And all is occurring without energy.
So the contraction doesn't require any
No.
ATP?
No. Yeah, really cool stuff. Now, there are some groups that are actually, and these are like physicists that are actually determining how much force is that DNA coming out with. As it's exiting, what's the force there? And they're finding that there's actually some initial force, as if you're overcrowding a space with DNA, and to allow for it to diffuse out, or the diffusion out of that icosahedral head is a result of you've just got so much DNA packed in there. And it also seems that the flow of that DNA out is actually regulated as well by enzymes or proteins that are at the end of that tube. So there seems to be some controlling in how quickly that DNA is moving out, which seems to have an effect on the regulation of gene expression.
So for the viruses that aren't actually entering the cell, once they've released their DNA, I'm guessing they just like degrade, they're done.
Yeah, I mean, at that point they're going to be free-floating, but yes, they are probably going to be chewed up by proteases at that point. It's just like a protein shell at that point. Yeah.
Why is it chewed up?
Well, all proteins are going to be degraded at some point, right? They're either going to denature, or proteases are going to get to them, and they're going to, or it's a combination of those two. That's what happens. And usually we don't concern ourselves with the actual viral structure after the DNA has gotten into the cell. I mean, if you wanted to really follow those, you probably could and figure out how long does it take for them to break down and lose their three-dimensional conformation. I don't really know the answer to that. Okay. Let me leave you with a thought then for us to pick up with not on Wednesday, because we've got an exam on Wednesday, but to pick up with again on Friday. We've just talked about the initial two steps here. We're going to move into the biosynthesis step. But one of the things that happens really early on, you've already seen this, the bacteriophage creates a hole in the bacterial cell. That's not a good thing. Right? So creating holes in the bacterial cell, no, that's not what I wanted to do. But this event right here, where we've actually got this attachment and then penetration is actually going to lead to a breakdown of the cell wall, which could end up leading to lysis. Lysis doesn't happen for another 20 to 30 minutes later, until the virus is ready to exit the cell. So how does it prevent lysis from occurring at such an early stage? Okay, and that's one of the things we'll talk about with biosynthesis. So see if you can find that. I'm guessing you may or may not find that information in the book. It may take a little bit of looking, but if you want, I've got your plates. Come and take a look. What I'm going to do today is I'm just going to pick an individual colony or individual plaque from each of them and just streak out a new plate of Pseudomonas and then take that plaque and kind of streak it out on the plate to get a really nice isolated one at this point. And then I'll have you guys start some cultures later in the week. But feel free to come and take a look at them if you want. Otherwise, have a great day.